So good morning, everyone. My name is Jennifer Jones. I am a research specialist here at the Illinois Soybean Association. Today we have Dr. Andrew Margenot and Heidi Asensio, and they will be presenting Navigating Soil Health Tests, Principles, Interpretation, and Utility. This webinar is being recorded. The recording, as well as information on ISA-funded research projects, can be found at ilsoyadvisor.com. All right, so Andrew, I'll have you go ahead and move to slide 2, where I can go ahead and introduce Andrew and Heidi. So we're pleased to have Dr. Andrew Margenot with us today. He is a soil scientist and associate professor at the University of Illinois in Urbana-Champaign. He joined the Illinois agricultural scene in 2017, where he leads a research team that evaluates nutrient biogeochemistry in our state and the greater north central US region. Dr. Margenot's research focuses on phosphorus management, soil health, and carbon crediting with the goal of supporting efficient use of nutrients for crop productivity that support environmental quality. We also have today Heidi. Heidi is a PhD student in the Department of Crop Sciences at the University of Illinois in Urbana-Champaign. She joined Dr. Margenot's lab in 2021. Her work seeks to establish and integrate benchmarks of soybean sustainability in Illinois using soil health, water quality, and climate-smart metrics. Heidi is passionate about collaborative work with farmers, understanding local agricultural practices, and ultimately generating knowledge to inform decision-making. All right, so we are very pleased to have Andrew and Heidi with us this morning, and I will go ahead and turn it over to you, Andrew, to introduce today's topic and begin your presentation.
Thanks, Jennifer, and thank you to ISA for helping us host this webinar and for hosting, and for helping us to put this together. So as you've seen from the emails and Twitter, this webinar is going to focus on a central theme of a ISA-funded project called the Benchmarking Project. More on that later. And we want to talk, Heidi and I, with you today about soil health, and specifically to provide our scientific opinion on how to go about categorizing these, how might one think about these, and how to interpret tests, including things like which tests could be used to guide how you manage, and simple questions that I think are important, which is, do we have confidence to the point of spending money on soil health testing? And here I want to emphasize that there's a lot of ongoing research. This is a very active field. And if there's one takeaway from today is that certainly we are getting better at measuring the biology of soils, and that is central to soil health. That makes it soil health, not quality, the predecessor term of the 1990s. But that said, there's a lot of basic work that needs to be done on validating these soil health tests on biology still, as well as developing, I think, new ones that get at what we know to be true from farmer experience, that having soils that differ in their biological activity, for lack of a better term, does make a difference for crop production and profitability. I'm not sure that we're there yet, but there's a lot of hope in getting there. So that's the main takeaway for today. Alrighty. So this, again, is part of a webinar series that our group is putting on with ISA that is centered on this project. It's a 5-year project. It's got 3 sites across the state, from Monmouth down to Ewing. And so we've got different soil types, and we're looking at tillage and cover crops in full factorials to understand a suite of outcomes. And these outcomes include things like climate change via greenhouse gas emissions, carbon credits as a result of that, profitability, of course, yield, soil health, and water quality via nutrient losses. So we want to integrate these different outcomes. Today's webinar then focuses on the top left, on the soil health concepts and theory of this multifaceted benchmarking project. Okay, so let's talk a bit more about soil health. Soil health assessments for soybean were covered in the first of this webinar series. And that's where we focused on this central question, which is, how does the practices that we use to manage soybean influence soil health, and vice versa? How does soil health influence soybean? So today we will answer 3 follow-up questions from that initial webinar that we gave on December 7th. And these include the practical outcomes that we want to get to today of what soil health indicators, meaning tests, should we be using? How should we sample, or when should we sample, better put? And the question that I think interests a lot of us, which is, do healthier soils, in fact, translate to more profitability for soybeans? And that can be done in two ways. It could be done as more yield for the same input costs, or, the increase in yield for the same input costs, or less input costs for the same yield. Both would, in theory, result in more profitability. Now, as a bit of background, the first webinar in this series was given on December 7th, and it was on soil health specific to soybeans. So in the soybean phase of the corn, soybean, or double crop with wheat crop rotation, what is known and what are emerging themes, and it's specific to the Midwest. So the YouTube link is here. We talk about the reciprocal relationship, soil health impact on soybeans, but also how soybeans and the system of management that we use to produce soybean can also benefit soil health. It's a two-way street here. So that's for your reference. I suggest checking that out if this topic interests you. Today, though, then the focus is on this first question. That webinar focused on this first question. We will deal with the other three. And to summarize the results of that first webinar as some background for today, we talked about how soybeans can be a very important lever to improve soil health. And as shown here, the four major principles of soil health management put forward by the NRCS listed here, we are able to hit these with different kinds of practices. So for example, cover cropping, which is more amenable to being in front of soybean than in front of corn for yield reasons. Well, that practice hits on the first of the four principles of USDA NRCS, and so forth, where we see that there's the ability of soybean to act as a lever. It's an entry point to introduce practices in the two-year crop rotation that benefit not just the soybean, but also the corn phase. Again, soybean is less wimpy than corn, so to speak, when it comes to cover cropping or no tillage and yield drags. And also double cropping with wheat raises the possibility of having roots and cover for most of the year, most of the months of the 24 months. So this was the main takeaway of the December 1st webinar. I want to come back to this and talk about then the three other questions that interest us. And these are the takeaways of the webinar. What should we be measuring? When should we be pulling soils to measure these soil health tests? And then this question, which is almost philosophical, I think, which is, do healthier soils mean greater profitability for soybean? So to get to these suggestions, because there aren't really hard answers, I don't think there will be. One, because it depends on context, but also at this moment in time, I don't think the science is there yet. It's developing. To get to suggestions on these three questions, we're going to talk about four things. First, a bit of background on soil health. I think knowing the origins is instructive to understand its limitations, but also where it could go. Two is the overall types and the theory behind soil health testing. Third, how do we begin to interpret soil health tests? And I'll focus on a couple case studies that illustrate the pitfalls, but also the potential. And finally, we'll end on this question of can, or better said, to what extent, a can is an if or black and white, but rather to what extent can soil health tests be used to guide how we manage systems, specifically soybeans? Part of this question, I think, is it worth testing for soil health? And there's a dollar sign there on purpose. Does it pay to test for soil health indicators that are being offered? Okay, so that's the game plan for this webinar. We'll end, of course, with this last question using a case study from this ISA benchmarking project that will be shown by Heidi, where she's been measuring after the first year of implementing conservation practices of no tillage and cover cropping, the outcomes for soil health. And that kind of gets at these questions of, is it worth testing and when should you begin to test? Is 1 year in sufficient? Let's begin with a brief history of soil health. This could itself be a 3-hour discussion, but to put it very succinctly, this graph here from Iowa State shows you the trajectory of research. This is the mention of the term soil health, soil quality, or soil health. So it's a research biased sampling when these were in highest frequency. So how commonly used were these going back to the 1900s? And as we can see, soil quality was the first term in the 1890s. Soil tilth took off, especially after the Great Dust Bowl. That's why we see it rocket after 1930 through the 1960s. Then it decreases by 1970. And then we see that there's an intersection of soil health and soil quality about 1990. And so for a while, the preferred term was soil quality. I think this is because that was contemporary with the Air and Water Quality Acts of the US EPA in the US, as well as in Europe. We were talking about quality of air, quality of waters. So there's also discussions at the time of should we also legislate or put standards on the quality of soils, but that's more from a contaminant perspective. Soil health then reared its head and really took off by the 1990s through today, and it displaced the term of soil quality. It was largely driven by Cornell in the early 2000s, putting a lot of weight behind for their New England Northeastern cropping systems, which are largely horticulture, dairy, a bit of silage corn, not great soils. They're smaller parcels. In that context, it was trying to understand soil health. So they did a lot of the initial work that we will be talking about. The thing that defines soil health from soil quality is by and large agreed to be the biology. So we have different ways to measure soil health, or let's say individual matrices that encompass it are what soil health is. But the addition and the emphasis on biology, be it earthworms to microbes, that is what by and large we think makes soil health health versus just quality. Now, if we go online to the NRCS website, there's a whole page dedicated to soil health. And the definition put forward by NRCS is the capacity of a soil to continue its function. And so it also pays tribute to that it's an ecosystem and it sustains not just humans, but also plants and animals, which are part of these functions. So function is very, very broad here, right? Function, by and large, we think of it, I think, in the ag world as food production, also fuel and fiber, but think also about things like holding water, not just for crops, but to hold the water to feed into the aquifer for drinking water. It's the foundation for homes. It's the materials for homes in some cases, like bricks. So it's a very broad definition, though I think by and large, most of the discussion is centered on food production, or let's say crop production more broadly. And then a more recent addition that was put a few years ago by NRCS as well was soil health can also be thought of as an assessment. So now it's less of this continued capacity to function, which I kind of like, even though it's kind of broad. It's more about an assessment, which would imply the process of exploring or measuring those functions. So these are a little bit not compatible, right? One is the function, one is the assessment of the function. But the point I think that they make well, the NRCS, is that it's these functions that will be sustained for a long time. Let's talk about then soil health testing. That's the main focus of today, after all. So let's talk about the types. The theory we've kind of touched on is the idea that we need more biology. We have to go beyond, say, the Melick test. And we know, for example, we've seen fields, I'm sure all of you have, where the Melick p-value, pound per acre is 10 pound per acre p, and we get great yields and we don't get responses to p. And people often propose, hey, that could be that there's a lot of active biological cycling of phosphorus. And so in that case, these agronomic chemical tests fall short. I wouldn't say that chemical, I think it gets really ragged on as a negative term. There's nothing wrong with chemical. Everything is a chemical. It is often used in a negative light. So when I say a chemical approach, I don't think that that's a bad thing. In fact, it's a very good thing because it gives us high confidence measurements of nutrients. So one of the conceptual theories of soil health testing is that you got to measure all the things. And all the things specifically refers to this tripartite, this holy trinity of physical, chemical, and biological. And on this left-hand figure, like the Olympic rings, we see there's different kinds of tests sorted by these. So this is the main approach currently for the theory, is that we have to measure health by the conjunction of physical, biological, and chemical. And that said, these things individually don't constitute health. It's their combination. This is what's called a latent trait. So here's a great example. Intelligence is a latent trait. We kind of know what intelligence is for a human, but there's no one good way to measure it, right? We know plenty of people who are street smart, but not book smart, and vice versa. I'm in academia. We're full of not very street smart people. This is an example of how there's no one metric, but it's this combination of things that collectively get at this underlying trait. So soul health is hard to measure directly as a latent trait. Okay. Now, there's two approaches overall for soul health testing. One is the indicators. These are the things that we measure, we can get a number on. And the other approach is how do we weigh these? How do we score these? And so these are two separate things. And of course, you need the indicator first to do the scoring. By and large, though, most folks at the moment are doing indicator-specific assessments. So, for example, you measure aggregate stability or SOM content, and you look at those individually without weighing or scoring them. And I'll talk about these in just a second. One note is that these are sometimes awkward categories. Aggregation, I think, is slept on in favor of biological stuff because it sounds, well, less sexy. And it's also mistaken as being physical. In some literal sense, aggregate stability is a physical thing, but it is the result of all three of these, right? You need good biology to build aggregates, and there has to be enough fertility there for the biology to be active. So I think aggregates shouldn't be put here. They should actually go in the center. They are, I think, the keystone. They are the central integrator indicator. But that's my opinion. Okay, so let's talk about the indicators first. These, again, are matrices that are measured. We get a hard number or just a number on it. And these are typically done in the lab, somewhere in the field, like infiltration rates. But we're talking 95% of these are done ex situ, out of place in the lab. Again, what the indicators that we oftentimes care about, so to speak, are the ones that are new, which are biological. If we flip back here, notice that the chemical ones are what we measure already. This is what we get as a soil test for fertility, right? We get macros and micros, pH. So in some sense, there's nothing new here for chemical. It's the biological, especially the more microbially centric indicators that are new. So I'm going to focus on those for the rest of today. Again, the concept here, and these are nice concepts, are integrating physical, chemical, and biological, different depictions of the same idea. And let's talk about how we got with these indicators, or how we got to these specific indicators. So back in 2019, there was a call by the USDA, NRCS, on it's time to standardize these indicators. So we've been doing soil health stuff, talking about it. Some groups like Cornell are proposing and doing their own thing. So the Fed said, let's try to centralize this. And so there was a call for input. This is a federal notice for comments in 2018, excuse me. And then if you dig into this document, we can categorize the proposed indicators that are biological as either processes or properties. This is the thing that we want to measure. This is a process or property that we know matters, and we try to approximate it by measuring an indicator. So this is an important disconnect, right? This is the symbol. This is the thing being symbolized in the world of art philosophy, for example. The painting is not of the individual. The individual is the individual. And we can broadly think about 6 categories in the 2018 NRCS call for inputs. Organic matter cycling, soil structure, microbe activity, the food for those microbes, how much N is crop available, and the microbe community diversity. These are the process or properties, and they would be measured through the proposed indicators. A lot of these you probably have heard about, like PLFA, labile or active carbon by epoxy. Respiration would probably go into that same category of number 4, etc. So now other institutes, about the same time, like the Soil Health Institute specifically in 2017, the year prior, they had 19 proposed indicators. I checked this morning, there's still the same proposed indicators. And again, we see the same tripartite categorization, physical, chemical, and biological. Now, what's what's I think nice about SHI here, the Institute, is that they have got tier 1 and tier 2 endorsed indicators. So tier 1 are things that we know can be measured with high confidence, basic quality control in the lab, and we know how to interpret them with high confidence. And that last part is a challenge for the biology. So think about things like Bray-P. That's pretty straightforward. That's a tier 1 indicator. Now, the tier 2, if we look at NRCS, are oftentimes contained as the recommended soil health biological assessments. As a side note, these are the documents to find the current USDA recommendations. Note 4503 and note 5406. 06 is for the infield stuff like aggregate stability, but just by the clod method, looking at roots, earthworm counts. These are very qualitative. These are more quantitative and all in the lab. So in the Soil Health Institute and in the NRCS documents, in different ways, the same thing is said, that the tier 2s, which are largely biological indicators, need a lot more work to understand what does this really mean. And meaning here is in a few senses. So one sense of what does this thing mean that we're measuring is how do you interpret the number? Is it, and especially if it goes up or down over time, is it a good or a bad thing? What is a good number to have? The other sense of meaning of the indicator is how do you act on it? One thing is to know if it's going up because there's more, say, activity by the microbes, but does that help you change how you manage for nitrogen? Those are different, albeit complementary questions. So let's look at the Soil Health Institute's tier 2s. These are, again, they need more research. This is in 2017. I'd argue it's still the same. All the ones, or let's say most of the ones that are tier 2 that need more work on what does it mean are all of the biological ones. Or I should correct that statement. They're largely all except respiration for the biological indicators. And specifically things like active carbon, POC that I will talk about, but also soil protein, which really isn't soil protein, by the way. It's a whole different discussion. It's just polyphenol content. And then things like PLFAs, which have been around for a long time, but they're really hard to interpret in both senses of what does it mean. So the biology that is newest to soil health is also the least understood. It kind of makes sense, right? It's the newest, it's more complex. But also this is one suggestion is be careful. At least I would be careful because I don't think we know with high confidence how to interpret these still ongoing research indicators. So those are the indicators, the things that we measure in a lab. It's a number on the test. Some labs or some individuals or consultants then will try to score them. So scoring is where you say, what is the good number? Well, you know, if you're looking at POC or at total respiration, what number do you want to be at and not be below? And so for this, as we can see from Cornell, it depends on sometimes the crop that you're growing, and sometimes it doesn't. So there's just one line for these. And the shapes are different. So there's some cases of more is better. Here for K, they have a more is better. So once you hit the certain level, this is the Morgan test. Those New Yorkers, they use Morgan, not Melick. And once you're at 100, it's okay to keep going. Or rather that kind of plateaus. If you think about phosphorus, because of the environmental loss risk above 25 ppm for the Morgan test, there's an optimum. So more is not better for the P. These are all chemical, by the way, right? And then pH, same thing, there's an optimum here. Some things like organic matter, enzyme activities, a lot of the biological ones put forward by NRCS, they are a more is better. The more microbes, the more carbon, the better. To my knowledge, not a single one that we just saw, if we go back here for just a second, not a single one of these has a optimum. More is always better. And that's kind of weird to me. I think that there probably are some trade-offs. If you've got, say, too much carbon, that might immobilize too much nitrate. So things to think about, excuse me. Okay, so this is the scoring. It tells you what is a good number. Issues with this is that what's your comparison? What's your sample size? What's your reference point? Is it native land use? So is it a prairie for our state? Is it a forest for New York State? Does it make sense to compare native land use versus ag? It's kind of weird. Should we look at 50 years of no-till and cover crops? But then what about soil type and texture? It's very important roles of texture on carbon, right? So it's not super clear. The other approach, separate from scoring, which tells you what's a good number, is averaging an indicator. And a good example here is shown on the right-hand side. So we've got a different approach, very nice visually, of what's a good number, again, based on these assumptions of what is good. Separately from that is you just kind of do a composite. So this is a simple, straightforward average done by the Cornell Soil Health Assessment, also known as the CASH. If you flip those words, the ordering, excuse me, and then just take a straight average. And that's interesting because now you're giving equal weight to the things that you picked as physical, biological, and chemical. I don't know how to go about this. So I'm not critiquing them. I don't know how to do this. Who's to say that we should weigh pH in the same way as respiration? There might be cases where we want to weigh the respiration more than the pH or vice versa. So these straight averages are kind of awkward because they are simply will be driven by the lowest number. These are pretty high numbers on some of these, but they're high in the physical and on the chemical. And so on the biological, not so great. So what if we did averages by each category? That would be interesting. The other approach not shown here is where you just have different weighing. You might weigh this 50% and these 25 and 25, or you even weigh one indicator 80% for some reason. So this is where there's completely a Wild West and there's no science behind any of the weighing. No science behind any of the weighing. I think that's where there's an opportunity to think about, if we know a system that functions well, can we retro-engineer, work back from a system that we know works as we want it to work, and then figure out what these indicator numbers are? And that might be a top-down way to score and then to weigh. Okay. So third topic for today is, how do we interpret these soil health tests? I've got 4 suggestions here. So number 1 is use the same lab, please, for your own sake. These soil tests are not reproducible, meaning if you send the same sample to a different lab, you will get a very different answer. This is some work by a colleague, Dr. Jordan Wade, when he was doing his master's at UC Davis, sent off same samples for respiration at 4 different labs. And to spare you all this, you get a completely different value on respiration depending on the lab that it goes to. I'd like to see data on, in the same lab, sending different sample or sending the same sample but marked as different samples. And as a side note, I'd always recommend, be it a MELIC test or a soil health test, do that with your labs. Tell them that they're different and see what you get back. It's good QC QA, quality control. So this kind of makes sense, I think, for 2 reasons. One is that biology is inherently noisy. It's hard to get, especially for these things like respiration consistent measures, because the process itself fluctuates. And second is that there's no regulations or quality control programs like the ALTA or the NAP program for, as we have for pH or for soil fertility testing. So there's no entity that goes around and has standard soils or reference soils or does blind checks on labs to ensure quality control. Right now, people just run the method as they want. And I can tell you, for things like active carbon, for respiration, every lab is different. And the problem is that that leads to non-comparable values. So if you're going to soil health tests, stick to the same lab and check that they've not changed the method over the time period that you sent them samples. Second is, be consistent in when you pull soils for soil health tests. This is some data here showing 3 indicators of N cycling enzymes and N pool. This is the soil protein pool being proposed by NRCS, and then the active carbon pool being proposed by the feds. And over a corn shown here, and then soybean with the cover crop here, 2-year phase. This is at Eric Miller's farm in Piatt County. We find that when you sample matters quite a bit. Different letters denote differences statistically. And so I think that we're seeing effects of the corn or the soybean or the cover crop roots. The more important point is that we don't see a difference among the 4 tillage treatments being shown here. They're all the same for a given time point, meaning that after 3 years of different tillage treatments, we saw no differences in these soil health tests, but we saw differences by time. So you're more sensitive to time in general than the treatment. We're finding that you have to go for sometimes 5 to 10 years for tillage treatments to see a difference, but that depends on the clay content that appears. So it's still the Wild West of when should you sample, how many years into a given practice going onto the land. But if you're going to test to track that progress, keep it consistent. Again, biology is maybe too sensitive. It's hair trigger sensitive to the outside world, temperature, moisture. So it's good to be consistent by date. Number three is be very cautious on how these tests are being interpreted, either in general or by the consultant or by the lab. We've talked about how the timing is essential. I think if interpretations don't caveat the timing, that's an issue. And then, sorry, folks, this is the repeat text here. I apologize for that. So to just say it to you orally, there's not a whole lot of data behind how to interpret these soil health tests with confidence beyond a general more is better. And I'm going to do a case study here on what the federal NRCS is calling active carbon. This is the permanganate oxidizable carbon, POXC. It's called active carbon, and I want to show you data that suggests it is neither active nor it's carbon that we're measuring. The principle is that we subject a soil sample to permanganate. It's used to sterilize drinking water. It burns up carbon if it's just carbon in the sample. And as you oxidize more of the permanganate, which is pink, you've got more carbon in the solution. Then you consume it, it gets less pink. So fainter color means more permanganate was reacted, which means that there was more carbon in the sample. That's the principle of the POX assay called active or labile carbon. A bit of background here. We've been using permanganates in research since the 1800s to measure total organic carbon. Cyril Hopkins at U of I was using it, and to get rid of lignin from coal because it's very lignin specific. Chew on that for a second. Lignin is not labile. It's, in fact, a polyphenolic poison made by plants to protect their tissues from fungal decomposition. It's why cellulosic ethanol has not come to fruition yet, because the lignin poisons the fermentation. You can ferment ethanol from corn grain, but not from the residues for this reason. So if permanganate is lignin specific, it's not really labile carbon. So there's already precedent for doubting this. There was a famous paper in 2003 that did a correlation of total carbon in soil with the POX-C value. And based on this, it was thought that this is active carbon. If that sounds a little thin to you for the evidence, it's because it is. There's no hard evidence ever put forward that POX-C was labile. It's sensitive to treatments like tillage, but that makes sense because we increase carbon under no-till, so that's just reflecting this correlation. It's not a causation, much less a mechanism of active. And it also changed to it also changes over the season, going back to be careful when you sample. But the thought was that, well, this reflects root dynamics, root exudates. That was the proposition put forward by Coleman. But again, these are all speculations. No one has actually shown that POX-C is a labile carbon. So we recently have been working on this problem or question, because I think it is a problem to not have evidence basis for telling folks what a soil test means. So in this first paper that we put out last year, that got us a lot of love from emails, was testing the basic assumption. Hey, is this POX-C fraction actually active carbon? And what we found was that the permanganate best reflects the amount of lignin in the soil sample. So we conclude that POX is not labile nor active, and here's why. We took things like cellulose, but also really good sugars, good for the microbe, but we would think of as microbial food, and then these very small compounds that are root exudates. But really, if we think that POX-C is labile carbon, it should be a high amount of reduction, so a high value should come for this measurement from the sugars. That would help us say, yeah, this is actually active carbon, it's microbial food. Instead, what we found was that there's very high value for ascorbic acid, vitamin C, right? I believe it is. So root exudates, okay, but look at lignin. It has the highest amount of POX-C value. And the lowest is actually for sugars. This is only 5 of 19 different compounds that we tested, by the way. We looked at amino acids, also microbial food. So what this means is that we cannot interpret POX-C as labile carbon. What's also kind of concerning is that we used the same amount of carbon in the sample and we got these different values. So there's a non-proportionate relationship of carbon in the sample with the amount of the permanganate reduced, and thus the POX-C value. But that is why then permanganate is very sensitive to what we call the functional groups, just the chemical composition, and specifically, it's preferentially going to get at lignin. So This might explain why we see higher epoxy values under no-till compared to chisel-till, because we've got more undecomposed lignin. That makes sense. So it's still useful as a proxy of the effect of how we manage, but why not just measure total carbon at that point? And we definitely cannot say that epoxy is labile or active. That's wrong. We also can't say that epoxy is carbon. So we found here that in this other paper, if we looked at the basic chemistry textbooks, I cracked it open, did some basic theory analysis, and realized that because of the assumptions in the stoichiometry, we're assuming a certain amount of carbon is oxidized by a certain amount of permanganate in the solution. And depending on the assumptions, the same amount of the permanganate can give you a wildly variable amount of carbon oxidized. So to spare you on the details, plus we're getting short on time, we cannot even measure carbon. This paper goes into all the details. It's open access. The point is that it's not even epoxy. It's just permanganate reduced. So that's a case study of be very careful. I would say soil protein has the same problem. Respiration has the least problem in terms of what are we actually measuring? How do we interpret that? So this is the final suggestion is it worth the price? Think about whether the test is paying for itself in terms of guiding increases in yield or decreases in inputs or both. Epoxy is going for at least 10 bucks. I'm seeing most labs now post-COVID, post-inflation, doing 25 per sample. Even these same labs that offer it say that it does not indicate better crop yield. I think that's a problem. We should have soil health tests that help us predict increased yields. That to me seems kind of important. Enzyme activities are also very expensive, and our lab does a lot of this work. We actually make the enzyme assays for NRCS to use, and I would never, ever recommend paying for an enzyme activity test. And I love enzymes. The science is not there yet. So final thing, and then Heidi will take the reins, is do healthier soils translate to more profitability? And the short answer is that currently we don't really know. We think this is a great hypothesis, right? We would expect to. We should retro-engineer soil health tests based on profitable systems with lower inputs. But we need more data. And the studies to date suggest this is not true. Currently offered soil health tests, current indicators do not relate to yield. They don't. Now, two ways to think about this. One is that more yield with the same input or lower input for the same yield, and you could argue both can happen. Two notes here. We can reduce inputs to a certain point. Once we are below the export of nutrients, the removal rates, we're now mining our soil. That can go on for some time if you've got stockpiled nutrients, but the system is going to crash. So when I say lower inputs, I mean the aspirational goal of only the maintenance rate. Second, we cannot make P and K. I hear a lot of stuff out there in soil health world about the biology can provide P and K. I've not put on P and K for 10 years. Yeah, you're mining your soils for P and K. N can be made from thin air, from BNF, but that's with legumes. And soybeans don't provide enough to cover what the corn needs. So just a couple of notes here that we should be careful and scientific about this. One example of how there's not much data, but it goes against soil health and yield. Some great work by Dr. Deanna Osmond in North Carolina, where she looked at across 3 sites, corn yield, and basically no correlation of these soil health tests or the index that Cornell uses, that score, with yield. I think that's an issue, right? Soil health tests should at least get at yield relationships. I'm going to skip through this and let Heidi talk about the results from our first year of this ISA benchmarking study to end on a nice example of how these can or may not be applied. So Heidi, take it from here, please.
Thank you, Dr. Margenot. Can we click next for the first slide? Yep. Perfect. So today I'll be sharing an update of the ISA-funded project titled Benchmarking and Integrating Soil Health, Water Quality, and Climate Smart Footprints of Illinois Soybeans, which will cover 4 cropping seasons from 2023 through 2026. We will focus on the response of different soil health indicators after 1 year of implementing the treatments of cover cropping and no-till at our experimental sites. And for this, the data that you'll hear about in this presentation is the result of soil analysis that were performed on samples collected in fall 2023. So the elongated shape of the state of Illinois allows for various climatic conditions that result in diverse soybean production systems. So in order to account for the differences between growing regions in the state, we established experimental plots in the Northwestern Illinois Agricultural Research and Demonstration Center facility, which is located near Monmouth in Warren County. We have one at the Crop Sciences Research and Education Center, which is located on the University of Illinois campus in Champaign County. And finally, at the Ewing Demonstration Center, which is located in Franklin County. So, as I mentioned before, our treatments consist of two well-established soil health management practices, which are cover cropping and no-till. We are testing tillage as conventional versus no-till, and as you can see in this picture, which was taken in Monmouth, we have a plot under our no-till treatment that has a little bit of soybean residue left over, and way in the back, you can see another plot with no-till where you can observe the corn residue left over. And for our cover cropping, we are testing cereal rye versus no cover crop. So in this final picture, we can observe the difference between a plot with the cereal rye here in the front and a plot without the cereal rye at our Urbana experimental site. Next, please. So after wrapping up the first cropping season of the experiment, we first wanted to know if soil health indicators responded differently to our cover crop and tillage treatments. Next, please. So the way that I like to think about the treatments that we're testing in this project is in the form of a soil conservation gradient. So basically, as we can see in this figure, we start with what we would consider, quote unquote, like a conventional practice of no cover cropping and tilling. And as we move further to the right, we see cover cropping plus till and no cover crop and no till sitting in the middle. Finally, in the other extreme of the gradient, we have what we could consider the most, quote unquote, conservative approach of having both cover crops and no till. So at our site in Monmouth, we have mollysols specifically from the OSCO series, and we are testing our results only in a conventional soybean corn rotation. Next, please. So the soil health indicators that we're testing can be divided into two groups. So we have permanganate oxidizable carbon and mineralizable carbon, which, as you heard from Andrew, we can, from the name also, they are related to soil carbon. And we also have a comprehensive assessment of different soil enzyme activities. We have enzymes involved in the carbon cycle, such as beta-glucosidase, or BG for short. We also have glucosaminidase, or NAG, which is involved in both the N and C cycles. We have leucinoaminopeptidase, LAP, which is mainly involved with nitrogen. And finally, we have phosphomonoesterase, or PME, which is involved in phosphorus cycling. Next, please. So our results showed that at Monmouth, activity of LAP and PME was different among treatments in our soybean phase. But how so? Next, please. When we take a closer look for the N-cycling enzyme LAP, we see significantly lower activity in the plots with no cover crops and tillage. Next, please. And similarly, in the case of the soil enzyme involved with phosphorus cycling, we again observed the lowest activity in the no cover crop tilled plots compared to our other treatments. Next, please. Perfect. If we travel towards central Illinois at Urbana, our experimental plots are located on mollysols of the Drummer series, and at our southernmost location at Ewing, we are working on alphisols from the Cisne series. Next, please. Now, something really exciting about this project is that at our central and southern sites, we are testing our treatments on both a conventional soybean corn rotation and on a double crop wheat, soybean, and corn rotation. Testing our treatments on both rotations ties up on how we're aiming to deliver results that are customized to the state's diverse soybean production systems. Next, please. So in this figure, we can see the same soil health indicators, but now we are evaluating them separately for each rotation. Next, please. So we found that after 1 year of implementing cover crops and no-till, there was no difference in soil health indicator response amongst treatments. Next, please. However, we still had a second question in mind. We now know that after 1 year, we did not see a different response from soil health indicators to cover cropping and till, but what about differences between rotations? How would soil health indicators in the soybean phase compare between the conventional rotation and a double crop rotation? And this is where things got a little bit more interesting. So at Urbana, we saw significant differences between rotations in the values of POC, permanganate oxidizable carbon, and leucine aminopeptidase activity. Next, please. So in the case of POC, we saw significantly higher values in the conventional rotation soybean compared to the double crop rotation soybean in the cover crop plus till and no cover crop plus till treatments. This is somewhat unexpected since there's a higher residue input in the double crop rotation, which is coming from the wheat. Nonetheless, as Dr. Margenot mentioned before, it is important to consider the nature of POC as an indicator and what it is actually measuring. Next, please. So in the case of LAP, we saw significantly higher activity with the double crop rotation in the no cover crop plus till and plus no till treatments compared to the conventional rotation. And just as a little bit side note of clarifying information, it might seem non-intuitive to have treatments labeled as cover crop and no cover crop when we have a double crop rotation system, because that wheat is acting as our cover crop. But since we have both phases, corn and wheat soybean, present at our sites each year, and the plots rotate between the crops, that means that one plot that might have the wheat and the soybean, the next year we'd have a cover crop before the corn. So we decided, and we're still figuring out what's the best way to present this, to keep those labels just to track how those changes are happening in the plots with our treatments. Next, please. In the case of Ewing, the soil health indicators that exhibited a significant difference between rotations were mineralizable carbon, NAG, and PME. Next, please. So mineralizable carbon was significantly higher in the soybean phase of the double crop rotation compared to the conventional under no cover crop and no till. And as you can see throughout on the graph, the double crop rotation had continuously higher values of mineralizable carbon, and this was to be expected since we have the extra residue incoming from that wheat before our soybean. Next, please. Our NAG activity was greater in the double crop rotation, especially in the cover crop plus no-till and the no cover crop plus till treatments. And next, please. Finally, PME was greater in our double crop rotations throughout our treatments, but it was significantly greater in the cover crop plus till treatment. So these are some of our findings after 1 year of work, and I am very much looking forward to sharing more about our results and years to come. And with this, I'm just going to check in if Dr. Margenot has some closing remarks, and thank you very much.
Thank you, Heidi. So I hope that was a nice way to finish this webinar. Some examples, and one thing that I'd like to note is the inconsistency of which indicator at what site in response to which practice responds most. Now that said, this is year one. One of the objectives of this benchmarking project, supported by ISA, is to understand what would be the right time to begin to look at soil health. We were sampling spring and fall for each of the treatments for each of 4 years at each of the 3 sites. So we hope to be able to identify whether there is a consistent soil health test that can pick up on what we know should be happening in terms of the soil health response to these practices across that gradient that Heidi had described. And then we've got great data on nutrient losses by leaching, greenhouse gas emissions, soil carbon buildup, and on yield, as well as profitability. And that will let us retro-engineer, using these same samples, potential additional soil health tests, not just the selection of profit versus carbon sequestration responsive indicators. Excuse me. So with that, I think we've got some time for questions. We'd be happy to take them. Thanks.
Thank you so much, Andrew and Heidi. That was an excellent presentation and just a lot of great information. So thank you so much. And audience, please feel free to drop any questions you have into the Q&A. I don't see any in there right now at the moment, but yeah, just Andrew. So I guess at this point in time, there's so much uncertainty. Things are still developing around these soil health metrics. What kind of recommendation would you give at this point to a farmer? Should they just put a pause on soil health sampling for the moment, or are there certain indicators that you would recommend in particular that they start with?
That's a great question. It might depend on how expensive these soil health tests. The way that I like to approach them is when they're comparable to a fertility test where you're getting Bray, P and K, etc., then it might be worthwhile. There's some packages that are very expensive, upwards of $60 per sample for soil health tests. Those I would be cautious about whether that value is there. Of all these tests, I think aggregate stability is the best, and that could be done at home. You can buy a simple mesh screen from Menards, and if you have a kitchen scale, you can weigh how much soil, once it's air dry, you put on top of the sieve. You slake it up and down like a metronome, 1 second up, 1 second down. So you dunk it 60 times, and then you measure the mass after it dries of what stayed on the sieve. This is basically what we do in the lab and charge $40 for. I think aggregate stability is the best integrator. It's a keystone of the chemical, physical, and biological. And fortunately, you do not need a lab to do pretty good measurements. So that would be my first suggestion, is do it yourself, because it's not hard. Second is respiration is probably good to get into for outsourcing. It's just incredibly sensitive to even how you wet the soil in the lab will drive the value of the CO2. So it's extremely sensitive to not just the lab, but who in the lab that day ran it. Studies have shown that it will change that number. Beyond that, I would actively suggest people do not consider Haney or these enzyme activities or soil protein or POC because the data is either not there to support it, and we're probably years away. B, they've been made up and there's no data to suggest that they are what we think they are. Or 3, there's data against them, including POC, which we've shown now from our lab is not even carbon. So if there's interest in measuring those, they might still tell you something about the direction of your system. Go for it. I'm not dissuading people from that, but just be very careful with what you're paying for what you're being told you're being given back as a measurement. There's one final test that I think is kind of neat. It's called the Soil My Undies test. And you heard me right. So you take cotton underwear and you bury it in the soil. This is basically an enzyme activity assay. So cotton is largely cellulose, 98% by mass. So when you bury underwear, the elastic tag lets you pull it back out. You can weigh the underwear before and after over a given time period. This is being used by the USDA. It began, I believe, in Iowa by the ARS folks there. And the percent change in weight, the decrease in weight over a unit of time, is a pretty good measurement of the overall decomposition activity. That's another do-at-home test. That's, a pack of underwear at the store.
Yeah, that's a pretty fun one too. Visually can really tell the difference just by looking at them, how the biology is different. So great.
Yes.
Thank you so much. I don't see any other questions coming in, so I think we'll go ahead and wrap it up for today. So thank you again so much to our presenters, Andrew and Heidi, for joining us. And thank you all for attending. Please stay tuned to illsoyadvisor.com for the recording of this webinar and for updates on this research project and other ISA-funded soybean production research. So thank you again and have a wonderful day.